The ASCEND trial was a multinational, double-blind, placebo-controlled clinical trial of pirfenidone for idiopathic pulmonary fibrosis. The trial enrolled 555 patients with mild to moderate IPF at numerous sites around the world. The primary endpoint was change in FVC at 52 weeks. The trial was well conducted with a high retention rate. The study hit its primary endpoint as measured by change in FVC of 10% or death at 1 year. There was a 48% relative risk reduction in that endpoint. And that endpoint was highly significant by a rank ANCOVA analysis with a p-value less than 0.001. That 48% relative risk reduction translates into a 15% absolute risk reduction in progression or death at 1 year. A 15% absolute risk reduction is a number needed to treat of 7. For every 7 people with mild to moderate idiopathic pulmonary fibrosis treated with pirfenidone for a year, we will save 1 of them from having progression or death by the end of that year. The study hit on multiple secondary endpoints as well. There was a reduction in the decline in 6-minute walk distance, and it hit on progression-free survival, which was a combined endpoint of death, decline in FVC by 10%, or a 50-meter decrement in 6-minute walk distance. The study had no detectable effect on shortness of breath, as measured by the UCSD Shortness of Breath Questionnaire, or on mortality. However, in a pooled analysis with the capacity data, in that combined analysis, there was a significant 45% relative risk reduction in the risk of death, which was highly significant. As expected, pirfenidone had some side effects associated with it, mostly GI side effects and rash. These were reported in a similar proportion of patients in the ASCEND trial as in the capacity trial in previous studies. Generally, the drug was well tolerated. So in summary, pirfenidone was shown to slow down progression and may have had a meaningful effect on the risk of death, at least for some patients. These results are really exciting, I think, for the whole community, and they're certainly consistent with what we've seen in prior studies, and we look forward to a lot of new data coming out over the next couple of years as we gain experience with this drug.
The Impasses trials have been presented at the ATS conference 2014 in San Diego, and these trials have been conducted in patients with IPF in order to test the hypothesis that nintedanib will be able to reduce disease progression in patients with IPF. Nintedanib is a tyrosine kinase inhibitor targeting VEGF, PDGF, and FGF, and all of these kinases have been shown to be involved in the pathogenesis of lung fibrosis in animal models and in ex vivo models. The results of a phase 2 trial that was published in the New England Journal of Medicine 2011 suggested that actually nintedanib at the dose of 150 milligrams twice a day may reduce disease progression and reduce the rate of acute exacerbation. Based on this preliminary data, the Impulses trial were designed. These are 2 identical, multinational, double-blind, placebo-controlled trials comparing nintedanib at 150 milligrams twice a day with placebo. The duration of the trials were 52 weeks, and the patients enrolled, the broad inclusion criteria were related to disease severity. In order to be enrolled in the Impulses trial, the patient had to have more than 50% predicted forced vital capacity with no upper limits, and the LCO has to be between 30 and 79% predicted. There were some other inclusion and exclusion criteria, but overall the population was representative of patients with IPF, and the baseline characteristics were balanced across the trials. There were more male. Average mean age was around 68 years. Most patients were former smoker or smokers, and the baseline FVC % predicted value was around 80% across all arms of the 2 trials. The primary endpoint was the annual rate of decline in forced vital capacity as measured in milliliter per year. And this primary endpoint was positive in both trials. And in both trials, nintedanib showed a reduction of 50% in the FVC annual rate of decline. Now, this primary analysis of the primary endpoint was supported by multiple secondary analyses in order to look at FVC decline in different ways, like change from baseline, or like categorical change of 5% and categorical change of 10%. And all of these secondary analyses on the primary endpoint were all supportive, and also when we looked at multiple ways of imputing missing data, still the size of the effect was robust and similar to the primary endpoint and primary analysis for both Impulses 1 and Impulses 2. One of the key secondary endpoints in the Impulses trials was the time to first acute exacerbation over 52 weeks of treatment. And for Impulses 2, nintedanib statistically significantly reduced the time to first acute exacerbation in patients treated with the drug, while in Impulses 1, the difference between placebo and nintedanib was not statistically significant. We have been trying to look in a predefined way in multiple analyses on the time to first acute exacerbation. One of these predefined analyses was the pooling of the two trials. Since the two trials were positive, we were able to pool the data for acute exacerbation, and we have seen a numerical reduction in the rate of acute exacerbation, but the p-value was not statistically significant. Another way of looking at acute exacerbation was to look at adjudicated acute exacerbation. In a predefined analysis, all events reported by the investigator as acute exacerbations were reviewed by an independent adjudication committee and were classified as confirmed acute exacerbation, suspected acute exacerbation, or not an acute exacerbation based on published pre-specified criteria. About 50% of the events reported by the investigator as acute exacerbation were reclassified as not acute exacerbation. So in a predefined pooled analysis of the acute exacerbation defined as confirmed or suspected, we did find a statistically significant reduction of the time to first acute exacerbation of about 70% in patients treated with nintedanib as compared to patients in placebo. Another key secondary endpoint was quality of life as measured by the St. George Respiratory Questionnaire. Impulses 2 showed a statistically significant reduction in deterioration of the quality of life in patients with IPF treated with nintedanib, while Impulses 1 did not provide the signal, and the pooled analysis was not statistically significant for a change in quality of life as measured by the St. George Respiratory Questionnaire. Mortality was another pre-specified endpoint in the Impulses trial, and although the trial were not powered to show a statistically significant effect on the reduction of mortality, however, looking at the all-cause mortality over 52 weeks, we have seen a numerical reduction in the number of deaths in patients treated with nintedanib as compared to patients treated with placebo, and that reduction was around 30%. In terms of side effects profile, as expected from the class and from the phase 2 data, the vast majority of the side effects were of gastrointestinal, with diarrhea being the most frequent side effects, with about 63% of patients in nintedanib as compared to 18% of patients in placebo reported at least one episode of diarrhea. Most of the episodes of diarrhea were mild to moderate, and less than 5% of these episodes resulted in drug discontinuation. So overall, we can say that for the first time we had two trials, randomized, controlled, placebo-controlled, international, that provided the same results in terms of primary endpoint. And these results were basically a reduction of 50% in disease progression as measured as the annual rate of decline in forced vital capacity. Nintedanib had side effects that were generally manageable and were also managed by dose reduction. And overall, I would say that these results support the concept that the treatment of patients with IPF with nintedanib results in reduction of disease progression.
I'm presenting the results of one of 3 large clinical trials reported at ATS this year, which is the second part of the IPF NET-sponsored PANTHER trial. The PANTHER studies was looking at 3 different arms, 2 different therapeutic approaches for IPF. One was a combined drug arm of prednisone, azathioprine, and high-dose acetylcysteine. The other was just high-dose acetylcysteine alone, both compared against a no therapy arm. Many of you may know that a couple of years ago the 3-dose arm was stopped early for no benefit and potential harm. So the report released this week was the kind of second half of this study, looking at NAC therapy alone compared to placebo. So this trial reported out on about 260 patients. They were equally randomized between high-dose acetylcysteine, that's 600 milligrams 3 times daily, and placebo, so about 130 patients in each arm. And the primary endpoint was change in forced vital capacity between baseline and week 52. The primary result of the study was no difference between the 2 arms in decline in forced vital capacity. There was a decline of 190 cc's in the placebo group and a decline of 180 cc's in the NAC group with a p-value of 0.8. There were 9 deaths and 6 acute exacerbations, so very small numbers, that's overall, and there was no difference between the 2 arms. And in general, secondary endpoints also were no different. There were a few secondary endpoints in select quality of life metrics which showed a benefit or potential benefit of NAC, but in general the results were no different between groups. In terms of side effects and adverse effects, there were really minimal side effects, but in terms of serious adverse events there were 2 of note. One was in the cardiac event category, where a little over 6% of patients in the NAC group had a serious cardiac event, whereas only about 2% of the placebo group did. And in the GI category, there were about 5% of patients in the placebo group who had a serious GI adverse event, and interestingly 0% in the high-dose NAC group. So the conclusion of the study and the conclusion in the article is that high-dose acetylcysteine used in patients with mild to moderate idiopathic pulmonary fibrosis has no benefit in slowing the rate of decline of forced vital capacity. There were several subgroup analyses reported. Some were pre-specified, including looking at severity of disease, presence of or coexisting emphysema. All of those showed no difference as well. However, the way this trial was conducted, as I mentioned earlier, included the cessation of the original 3-drug arm, and at that time there was a pause in the protocol and kind of a bit of a reconstruction of the protocol before it was resumed. And so post hoc there was an analysis done looking at the pre-alert or pre-stopping and post-alert or post-stopping patients, so patients who were enrolled before or after that date. And when that's done, there is at least suggestion that there may be some differential effect in those 2 groups. Importantly, the statistical comparison of the pre-alert and post-alert groups show that there is no difference between those 2 cohorts, both in baseline characteristics and in response to NAC. And there is no evidence of an interaction of the alert period with the primary endpoint or treatment effect. In conclusion, this trial shows no benefit of high-dose acetylcysteine in the treatment of mild to moderate idiopathic pulmonary fibrosis.